Hello friends, welcome back to our YouTube channel Doctor's Corner. In this today's lecture, we are going to discuss about an important topic in renal physiology, that is transport maximum for glucose. Okay, so this transport maximum for glucose, it is also termed as TMG. So similar concept of TMG, there is one more concept, that is renal threshold for glucose. So what is the renal threshold for glucose? And one more concept is about splay in the curve of transport maximum reabsorption curve. Okay, so basically we are going to discuss this graph today. Okay, so this can be an important theory question in the subject physiology related to the filtration, excretion and reabsorption of the glucose across the nephrons. Okay, now to understand this, we'll discuss some basic concepts. Okay, some basic concept how exactly the nephrons will work, how there will be the reabsorption, excretion and filtration, and how more specifically the glucose is reabsorbed. Okay, so clinically it is related to glucoseuria, that is appearance of the glucose in the urine. Normally in a healthy person there is no glucose in the urine. Okay, so the glucose will be absent in the urine, but in clinical condition like diabetes mellitus, where the blood glucose level is high, the urine also consists of the glucose, that is there is appearance of the glucose in the urine and it can be detected by various tests. Okay, so why this glucose will appear in the urine and what is the mechanism we are going to discuss today. So we'll come to this today's topic. First we'll have some background about the functioning of the nephron. So we'll discuss four important concepts first, that is for the formation of the urine across the nephron, that is the filtration process will be there. So there are four processes. So one process is the filtration, second is the excretion, third is about the reabsorption, and finally secretion. So what exactly is this filtration, excretion, reabsorption and secretion with respect to nephron? Let us discuss. So as you will be aware, human kidneys will be having around 1 million nephrons in each kidneys, 1 to 1.1 million nephrons. So totally we will have two kidneys, so it means around 2 to 2.2 million nephrons totally in both the kidneys. Okay, so I'll just draw a line diagram showing the tubular structures of the nephron. Okay, so I hope everyone knows that there is a Bowman's capsule here, and continuation of that Bowman's capsule will be the proximal convoluted tubule. So I'm drawing just the tubular structures, proximal convoluted tubule. Then we have a bend that is known as loop of Henle. Okay, loop of Henle, just I'm drawing a line diagram, not a correct anatomical diagram. Then we have the distal convoluted tubules, then finally the collecting duct. Okay, and ultimately it will drain the urine. So basically there are glomerular capillaries. Okay, there is one blood capillaries which are present, glomerular capillary tuft, which has an efferent arteriole. Efferent arteriole is a structure from which the blood will be coming to this nephrons, and there is something known as efferent arteriole. Okay, so these are the basically capillaries. So through these capillaries ultimately there is filtration that takes place. Okay, filtration that takes place, that is this first concept. What is filtration? Okay, filtration that takes place. So what are the substances that are filtered? Sodium will get filtered, chloride, glucose, okay, bicarbonate, even water gets filtered. How much filtration takes place normally? It is around 125 ml per minute. Okay, it is around 125 ml per minute, and this it is known as glomerular filtration rate. Okay, we are not going to go in detail about this thing, just know glomerular filtration rate, that is the filtration rate at the level of the glomerulus. So this total plasma, that is the liquid part of the blood, gets filtered. Almost everything gets filtered except for the plasma proteins. Okay, so this gets filtered. So this filtration takes place at the rate of 125 ml per minute, or if we see in the total 24 hours duration, that is in one day, it will be around 180 liters per minute. That is in both the kidneys the blood plasma will be filtered at a rate of 180 liters. Just imagine how much will be the 180 liters. 180 liters are filtered in each day in both the kidneys in normal adult. Okay, now after this filtration, 180 out of that 180 liters, only around, on an average, around 1.5 liters per day is excreted via urine. Okay, the urine which is formed by the nephrons of our kidneys, ultimately out of 180 liters which is filtered, only 1.5 liters. That means, so this excretion of 1.5 liter is called as excretion. So we have discussed what is filtration, what is excretion. Okay, filtration is nothing but this filtration, GFR. Then excretion is the formation of urine. Okay, so it is around 1.5 liters per day. So it is average. Okay, it will, however, it will vary in range. Some will have 2.5 liters per day. So if it is more than 3.5 liters, it is termed as polyuria. It is increased urine excretion. Okay, now this efferent arteriole further continues into one more group of arterioles. These are known as peritubular capillaries. Why they are termed as peritubular? Because they are just near to the tubule. So this is known as peritubular capillaries. Okay, so here you can see capillaries will continue as capillaries. Okay, efferent arteriole continues as the peritubular capillaries. Now, as you can see, as I said, 180 liters is filtered through this, but only 1.5 liters is excreted as urine. Okay, this is I am showing just one nephron, but it is collectively from all the millions nephrons from both the kidneys. Okay, so now out of that 180 liters, if 1.5 liters is excreted per day, just theoretically, so around 178.5 liters per day will be reabsorbed back into the blood or the interstitial fluid. Now, this process of reabsorption of the filtered plasma back into the blood vessel, this is known as simply reabsorption. Okay, so what we have discussed: filtration, excretion, and reabsorption. Okay, so maximum amount of these substances are reabsorbed in the proximal convoluted tubule, that is PCT. This is loop of Henle. Then we have the distal convoluted tubule, then collecting duct or collecting tubules. Okay, so maximum amount it is filtered in the PCT. So for example, sodium and all, so 65% is filtered here, and glucose, that is the today's topic, almost 100% of the glucose which is filtered through this is 100%, almost 100% of the glucose is filtered through this glomerular membrane, is reabsorbed back into this blood vessels or the interstitial. Okay, interstitial fluid, and literally there will be nil glucose in the urine in a normal person who is having a normal blood glucose level. Are you following? For example, if on an average a person is having 100 milligram per 100 ml of the blood, that 100 milligram will be filtered through this glomerular filtration membrane, and ultimately all that 100 milligram is literally reabsorbed, and there is no glucose will be present normally in the urine. That is the basic concept. Okay, this is this one. Then there is one more term known as secretion. So few substances from the either from the interstitial fluid or from the blood direct enters back into the proximal convoluted tubule or into the distal convoluted tubule. So this process, it is known as secretion. Okay, I hope this four basic concept is clear. What is filtration, what is excretion, what is reabsorption, what is secretion with respect to the nephrons and formation of the urine. Now, having these concepts in mind, let us now discuss the transport of glucose. Very important. Okay, some basic about the transport of glucose, then we'll come to our basic topic, that is the transport maximum of glucose, and this graph, because to understand this graph we should have a knowledge of what is filtration, what is excretion, what is reabsorption. Okay, now how glucose is transported at the level of the nephron we are going to discuss, that is transport of glucose from the proximal convoluted tubule. Okay, so let me draw a bigger diagram of this one, proximal, this first Bowman's capsule. Okay, this is a Bowman's capsule, and we have the PCT, proximal convoluted tubule. Okay, and we are having a blood vessels, glomerular capillaries, that is this glomerular, this is efferent arteriole, this is efferent arteriole, and there is a filtration of the glucose. I will draw this glucose with the blue color. These are the glucose molecules. Okay, So how they are filtered, and here we have the peritubular capillaries. Okay, peritubular capillaries which are continuous from this efferent arteriole. Okay, now these glucose molecules that is we have described now, which gets filtered, will ultimately reabsorbed back into the peritubular capillaries. How they are reabsorbed back is, so there is some mechanisms and they will be reabsorbed from this epithelial cells of the proximal convoluted tubule. Okay, so this epithelial cells of the proximal convoluted tubule, there are certain transport proteins which are present on the membranes of this epithelial cells, and they will drag the glucose into this interstitial or the blood vessels, and almost 100% is dragged. Okay, so what are that transport proteins and all we'll discuss. So I will enlarge this part now. I will enlarge this part. This is the luminal part, that is the lumen of the PCT, luminal part. This is the interstitial part, interstitial fluid, and this is the peritubular capillaries. I will enlarge just one or two cells from this proximal convoluted tubule here to have a better understanding. Okay, I'll draw that cells here. Now this, imagine this bigger cell, and what I'm drawing is the one of the epithelial cells of the PCT, proximal convoluted tubule. Okay, there is a one more epithelial cell here. Okay, one more epithelial cell will be there. Okay, so this epithelial cells will be there. These are epithelial cells. Okay, now these are the lumen of proximal convoluted tubule, and here we have the peritubular capillaries or the blood vessels, that is interstitial site. Okay, now this epithelial cells on the luminal surface, they will have a brush border. Okay, they will have an appearance of a brush border. So let me draw that brush border thing like this. So how they will be, so it will be like this brush border type, like microvilli. Okay, so this surface, this is known as basolateral membrane. What is this known as? Basolateral membrane, basolateral part or the basolateral membrane of the epithelial cells of the proximal convoluted tubule. Okay, hope you are understanding this concept. Okay, and this is the apical surface membrane. Okay, which has the microvilli. Now on this basolateral membrane of this proximal proximal convoluted tubule, there are certain transporters present, protein transporters, proteins. This protein transporter, it is known as sodium potassium pump, or simply it is known as sodium potassium ATPase. Hope you know this thing, sodium potassium ATPase. Okay, these transporters which I have marked with green color. So what is the function of this sodium potassium pump or sodium potassium ATPase is, simply they will take away 3 sodium molecules from inside the cell to the exterior, and they will in turn transfer 2 potassium ions inside, from outside to inside. Okay, so ultimately there is loss of one negative one positive charge. So interior of the cell will be negative, that is around minus 70 millivolts. So continuously they will be transporting or pumping these 3 sodium ions from inside the cell to outside, and 2 potassium ions from outside the cell to inside the cell. Okay, so this is type of transport which is against the concentration gradient and requires the energy with the help of ATP, which is generated by the mitochondria which are present in the cell. Okay, this type of transport is simply known as primary transport. Okay, hope general physiology you might have studied what is transports. Okay, we are going to discuss about the secondary active transport also. This is primary active transport. Active transport means which requires energy and there is movement of the substances against the concentration gradient. Okay, so against the concentration gradient, the 3 sodium ions are moving outside. Okay, and 2 potassium ions are moving inside. So this, with the help of energy, is known as primary active transport. Okay, hope this concept is clear. Now, because this sodium potassium pump is continuously working, there is a deficiency of the sodium ions inside the cell, and in the lumen, as the glomerular filtration is going on, there are sodium ions will be present here. They are also filtered there. So these sodium ions as there is a electrical gradient as well as concentration gradient. Electrical gradient because there is negative charges are more, positive charges are going on, that is because of that electrical gradient, and concentration gradient because sodium ions are less here. The sodium ions will enter inside this cell. Now sodium is a charged particle, it can't directly diffuse inside, it requires certain carrier proteins. Okay. So with the help of this carrier protein, sodium ion will enter inside this cell. Okay. Now this carrier protein not only transport the sodium inside the cell, along with this it will transport the glucose molecule. It will transport the glucose molecule along with the sodium. This is the glucose molecule with the blue color thing which is present in the lumen of the proximal convoluted tubule. This is called as co-transport, and the name of this transporter is known as sodium dependent glucose co-transporter. It is termed as SGLT. Sodium dependent glucose transporter. More specifically, it is SGLT2 in the initial part of the proximal convoluted tubule, which is present on the brush border side. It is on the luminal surface, not on the basolateral side. Basolateral side we have primary active transport by the sodium potassium pump. Now because of this pump only, sodium is coming inside. That is, the energy which was generated by this primary active transport causes the transport of the sodium, and secondarily the transport of the glucose. So this transport by this SGLT is known as secondary active transport. Hope the concept is clear. Here you are seeing on the same cell, on one side there is a primary active transport, on the another side what you are seeing is the secondary active transport. Okay. So secondary active transport means it will not it is an active transport, okay, but the energy utilized is by this some other transporter protein, not from this directly. Suppose if there is this is not functioning, say sodium potassium pump is blocked by some certain substances, okay, then this sodium will not enter and secondarily glucose will not enter. For this glucose to get entered inside the cell, this has to function. So this is known as secondary active transport of the glucose molecule along with the sodium. This is sodium co-transport, okay, and this is secondary active transport, right? So after entering inside the cell, glucose molecule will now go outside. It will go outside again with the help of one more carrier protein. It will diffuse outside. And ultimately enter into the interstitial fluid. And this carrier protein which will diffuse, that is the facilitated diffusion, which will diffuse this glucose from this basolateral membrane outside into the interstitial fluid and ultimately into the blood vessels, that is known as glucose transporter or termed as GLUT molecule. Okay, here it is GLUT number 2, okay, in the proximal convoluted tubule. So here on the proximal convoluted tubule epithelial cells, okay, here where is this one? This one, proximal convoluted tubule epithelial cell. This is a nephron. Nephron has glomerulus, Bowman's capsule, proximal convoluted tubule. Proximal convoluted tubule epithelial cells we have enlarged. So on epithelial cells, on the basolateral membrane we have sodium potassium pump, and on the brush border cells we have sodium dependent glucose co-transporter. That is secondary active transport. This is primary active transport, and once the glucose enters here, the glucose will get diffused outside this cell into the interstitial fluid and ultimately into the blood, taken back. It is by GLUT transporter, that is the glucose transporter number 2. Okay, this is how glucose is transported at the proximal convoluted tubule. And just let me remind you, almost 100% of the glucose which is filtered out will get reabsorbed in the proximal convoluted tubule. That's why in a normal person there is no glucose is seen in the urine. Okay, so almost nil, practically nil, because glucose is a very important substance required for the metabolism and energy. So we can't lose this glucose into the urine. So whichever glucose is found which is filtered from the blood, for example 100 molecules of the glucose is filtered through this blood vessels, okay, through the glomerular capillaries into the Bowman's capsule, that 100 molecules will get reabsorbed back in a healthy individual. Now when it will come into the urine, what will it will cause us to come into the urine, especially in diabetes mellitus where the blood sugar level is more, how exactly it will come into the urine. Let us discuss with the help of a graph. Okay, so I'll just draw a graph. So this is what we are going to discuss. Today's topic is transport maximum. Okay, this is a ready-made graph, but I will draw a graph for you people so that the concept will be more clear. So let me draw a graph. So hope you are understanding this. I think the video is a bit lengthy, but still it's worth to understand this basics. Okay, now on the x-axis I am going to mention the plasma glucose level. Okay, it is the blood glucose level, plasma glucose level, and the units will be milligram per 100 ml. 100 ml means deciliter, milligram per deciliter. Okay, so I will start from 0, 100, 200, 300, 400, 500. On the y-axis, I will be drawing the filtration rate of this glucose, excretion rate, as well as the reabsorption rate. Rate means milligram per minute. Okay, so with what I will be doing, filtration rate, then reabsorption rate, and finally excretion rate. Okay, by 3 graphs we are going to draw. Okay, again I will be drawing this 100 milligram per minute, 200 milligram per minute, 300, 400, 500, whatever about that also. Now first coming to the filtration. Okay, as you can see in this ready-made graph, this pink color line, there is a linear relationship. Okay, I'll just draw it with the again with the pink color only here to make the concept more clear. So see, pink or purple color. See, if the blood glucose level is 100 milligram, then the filtration means at the level of the glomerulus, so it will be 100 milligram. If it is 200 milligram, again it is 200 milligram. If it is 3, 400, 500, 500. So it has a linear relationship. This is filtration, graph for filtration. Okay, what is this filtration? We have already discussed. Again I will just make you a rough diagram of the nephron and make you try to understand it. So this is a rough diagram of nephron. So what exactly I am going to show you is, this is a blood vessel, it is glomerulus, afferent arteriole, then efferent arteriole. So ultimately the glucose molecule, okay, glucose molecules are filtered here from this blood to here inside. Suppose there is 100 milligram per deciliter of the glucose which is filtered here. Okay, so 100 is gram is that all the glucose molecules are filtered here, 100 milligram. Suppose there are 200 milligram will enter here. Suppose there is 300 milligram of the glucose will enter here. That is, there is a linear relationship. Okay, the linear relationship between the plasma glucose level and the filtration rate, that is milligram per minute. Okay, that is the linear relation filtration. Now coming to the reabsorption, that is after filtration, this will enter into the peritubular capillaries, that is the blood vessels, this glucose molecule. So we have discussed how exactly with the help of the primary and secondary active transport ultimately it will come from this proximal convoluted tubule cells. Just now we have discussed in detail about this. So it is now reabsorbed almost 100%. But if the blood glucose level is more than 200, more than 300, then it will not reabsorb all the glucose. Why? Because the carrier proteins, so now we have discussed these carrier proteins, that is this sodium glucose transporters, these get saturated, that is they have the capacity just to carry around 100 or 200 glucose molecules. If there is 300 glucose molecules or 400 glucose molecules or 400 milligram per deciliter, then it may not absorb or reabsorb all of the glucose back into the blood. So some amount of the glucose now will come into the urine. Okay, so let us draw the reabsorption graph. So this is blue color line is the reabsorption graph. See, it is not as linear as the filtration, that was straight. So when the blood glucose level is up to 200, it is straight. After that you can see there is a slight bend, and around 375 it becomes exactly straight here. Okay, so let me draw here for you with the help of the blue color line. So see, when the blood glucose level is 100 milligram, the reabsorption is 100. Up to 200 there is a linear line. Okay, up to 200 is a linear line reabsorption. Same like filtration. Whatever it is filtered, everything is absorbed. But after that, you can see there is a curve, and after around 375 milligram, it will again become a straight line. Okay, it will become a straight line. It will not become abruptly straight line. There is a curve. This curve, it is known as splay. We are going to discuss this. What is this splay? Right now, concentrate on the reabsorption. So at the level of around 375 mg of blood glucose level, there is no reabsorption seen. That is, whatever glucose enters into the lumen after 375 mg, that all will be gone through urine. Okay, so after 375 mg of the blood glucose level, whatever this will come, it will be gone direct into the urine. Okay, it will come into the urine. Till 375, the glucose transporters can absorb. Okay, that is known as transport maximum. So what is transport maximum? Is the maximum rate of reabsorption, okay, of the glucose, after which the urine is glucose is not transported and the glucose molecule will appear in the urine. That is known as transport maximum. Why there will be transport maximum? Because for the transportation of the glucose, the carrier protein has certain limit. That is the maximum limit. After that, there is no transportation or no reabsorption takes place, and the glucose molecules simply go out because there is no carrier proteins available. They all are saturated. That is reabsorption curve. Okay. Now, but practically what we see is at around 200 milligram of the arterial plasma, the glucose will start appearing in the urine. Okay, as you can see in this ready-made graph, this green color line. After 200 milligram of the plasma, slowly they will start appearing in the urine. That is the excretion. Okay, I'll draw it for you. Then, so see, after 200 ml, it will start appearing, and after that it will become linear. The line becomes parallel with the filtration graph, and this green color line is the excretion curve. Excretion curve. Okay, excretion curve. Excretion means appearing in the urine. So in the urine, the blood glucose level will not be up to 200 mg. There will be no glucose will be present in the urine by the detectable test. But after 200 milligram, if the blood glucose level is 200 milligram, for the first time, in the urine, if the glucose is appearing, then that term it is called as renal threshold. That is known as renal threshold for blood glucose. What is renal threshold? So the blood glucose level at which the glucose appears for the first time in the urine, it is around 200 milligram per 100 ml in the arterial plasma, and if we consider the same in the venous blood, then it is around 180 milligram. That is the renal threshold. That is the blood glucose level at which the glucose will appear in the urine for the first time. Okay, but here what does our common sense says is, when the maximum all the transport proteins are utilized, that is transport maximum, at that level it should appear in the urine. But why the glucose is appearing before only? Okay, why there is a deviation from this? That is known as the splay. That is the deviation from this renal threshold to the transport maximum. So why the difference is, there are millions of nephrons in our kidney, and each of these nephrons will have different transport proteins. Okay, different transport proteins. Few nephrons will have more transport proteins. This one SGLT, glucose transporters, GLUT transporters, sodium potassium pump. Few will have less or more. Okay, so suppose few nephrons got saturated early, that is by 200 milligram only they got saturated, and after 200 milligram the glucose will not reabsorb in that nephrons, and they will start excreting the glucose in the urine, and that is the reason for the renal threshold. Okay, first time they will appear, but still few nephrons will be functioning and they will be trying to reabsorb, but at the level of 375 milligram, all the nephrons, that is the maximum limit, will get saturated, and after that whatever glucose is coming, they can't be reabsorbed and they will appear in the urine, or they get excreted, and they have a parallel line with the filtration, okay, after 375 mg, okay, they will be having a parallel line excretion with the filtered, that is whatever it is filtered, capacity is saturated, so everything it is coming into the urine, okay. So that is the relationship, and you can see there is in this reabsorption curve, it is blue color line, so it is not sharp bend like this, there is a certain curve initially, because different nephrons will act differently, so that's the cause for the renal splay. Okay, one more reason for the renal splay is, one is different nephrons, a different capacity to handle the reabsorb the glucose. Another thing is these glucose transporters, SGLT, so this sodium binding or the glucose binding affinity is less, okay, so few affinities, due to that also it will vary, so there is a variation between the different nephrons, that is all nephrons won't work in an equal manner, okay. So hope you understand this today's topic. So what we have discussed today, we have discussed about the transport maximum of the glucose, that is around 375 milligram per 100 ml in males, and in females it is around 300 or 350 milligram. Renal threshold in the venous blood, it is around 180 milligram per deciliter or 100 ml, or from the arterial plasma it is 200 milligram per deciliter, and renal splay is the deviation between this renal threshold and the transport maximum glucose. Okay, hope the concept is clear. So if you liked our video, please share this link with your friends and do subscribe our channel. Thank you.